For thousands of years, if you looked at a decaying log or a patch of wet mud, the origin of life seemed obvious. People saw maggots appearing on spoiled meat, or frogs crawling out of the riverbank, and concluded that living creatures simply materialized out of non-living matter. This concept was known as abiogenesis. Abiogenesis is the theory that biological life arises spontaneously from inorganic materials. Dating back to Aristotle in the 4th century BC, this idea served as the unchallenged philosophical dogma for how the natural world operated. In the 1590s, the catalyst for a scientific shift arrived from Dutch glassmakers who figured out how to curve lenses and stack them inside a tube. This invention allowed them to magnify objects up to 20 times their normal size. By bending light through a piece of polished glass, early researchers could suddenly see physical details that had always been invisible to the naked eye. This optical trick challenged humanity's oldest assumptions about how life begins. In 1665, an English scientist named Robert Hooke pointed one of these early microscopes at a thin slice of cork, the bark of a tree. Because he didn't have electric light, he used a flame and a water globe to focus beams onto his specimen. What Hooke saw under the lens was a rigid, geometric grid of tiny, empty chambers. Because they reminded him of the sparse dormitory rooms where monks lived in monasteries, he called them cellula, giving us the modern word cell. But Hooke was actually looking at a graveyard. The cork was made of dead plant tissue, meaning those empty honeycomb structures were just the stiff outer walls of the plant. He couldn't see any of the internal liquids or biological mechanics in linen chew. Hooke's compound microscope magnified objects by about 20 times. Today, modern equipment like the electron microscope can magnify specimens by millions of times. This jump in resolution reveals the microscopic biological activity that early researchers could not yet perceive. A few years later, in the 1670s, another Dutch lensmaker named Antony van Leeuwenhoek started grinding his own glass with different curvatures. He built a microscope that magnified objects by 270 times, using it to inspect drops of pond water and the plaque from his own teeth. Inside those water drops, he saw living, moving creatures swimming erratically across his slide. He called them animalcules, recording the first human observation of single-celled organisms actually going about the business of staying alive. Observations continued to pile up over the next century and a half, until 1838. A German botanist named Matthias Schleiden, who spent his days examining plant tissue, sat down with physiologist Theodor Schwann, who was looking at the nervous systems of animals. As they compared notes, they realized that whether they were looking at a maple tree or a cat, the microscopic architecture was identical. Both the complex plant and the complex animal were constructed from the exact same biological base units. This realization established the first two tenets of classical cell theory. First, all living organisms are composed of cells, and second, the cell is the basic structural and functional unit of life. Even with those two rules established, the 19th century scientific community remained divided over a lingering question. They knew living things were made of cells, but they still didn't know how those cells originally got there. Scientists like Robert Remak and Rudolf Virchow argued that cells reproduced by dividing. But a large faction of the old guard pushed back, insisting that abiogenesis was still happening, that invisible components in the air were combining with non-living matter to spontaneously generate microscopic life. In the 1860s, the French scientist Louis Pasteur designed an experiment to test this exact claim. He set out to show that the air itself was carrying existing microbes and that life was not appearing from non-living matter. Pasteur filled a glass flask with broth and boiled it to kill off existing bacteria. He stretched the neck of the glass into a long, S-shaped curve. This specific shape allowed outside air to flow freely in, but gravity forced airborne dust to settle in the lowest dip, keeping microbes far away from the liquid. Days and weeks passed, and the broth remained completely clear. Without physical contact with existing microbes, nothing grew. Pasteur's result provided the final proof for the third tenet of classical cell theory. All cells arise only from pre-existing cells. This discovery effectively replaced the dogma of abiogenesis with a law based on observation. Modern biology has shifted to understanding how these microscopic units build complex creatures. That process starts with something called a totipotent stem cell. A totipotent cell is a generalized base unit that has no specific job yet, but holds the potential to divide and develop into any specialized cell type in the body. As an organism develops, these generalized cells multiply and start taking on specific roles. They change their structure and physically lock together to form tissue. In the case of muscle tissue, they arrange themselves into tightly packed, linear bundles designed to contract and generate physical force. Biology then scales up another level. Different types of specialized tissues, like muscle layers, connective webs, and protective linings, group together to form distinct organs, like the stomach. From there, individual organs collaborate to execute large-scale functions. The stomach coordinates with the liver, pancreas, and intestines to form the complete digestive system. This hierarchy is known as the division of labor. By allowing specific cells to handle specific tasks like digestion or respiration, multicellular organisms can survive and operate far more efficiently than if every single cell had to do everything on its own. Over the last century, electron microscopes and genetic mapping have allowed us to see even deeper into the chemical processes of life. Because of this, classical cell theory has been updated with a few modern rules. We now know that when cells divide, they pass down their exact genetic blueprints in the form of DNA. We also know that the flow of chemical energy, the actual metabolism keeping an organism alive, happens strictly within the boundaries of the cells themselves. This definition excludes viruses. Viruses carry their genetic information as RNA, a single-stranded molecule containing genetic code, and they cannot reproduce or metabolize energy on their own. They must hijack a host cell to do the work for them, which is why biologists classify them as a biological exception. The human body is constructed from roughly 100 trillion of these individual units. We are walking cities of specialized cells, operating a vast internal economy that remained hidden until humanity learned to bend light through glass.